Hey everyone, this is our lesson on mitosis, and by the end of this lesson, you should be able to analyze how a cell grows and reproduces in terms of interphase, mitosis, and cytokinesis. So first, let's talk about how cells grow and divide. So they go through what's called the cell cycle, and there are 3 periods of this cell cycle in reproduction. So the first one is growth, and this is interphase. This is the phase of the cell cycle that the cells spend the most time in. They are in interphase most of their life. Division of the nucleus, which is mitosis, which is reproduction of the cells. And during this, obviously, the nucleus divides. And then the division of the cytoplasm, which is what actually causes the entire cell to split into 2 separate cells, which is cytokinesis. So let's talk about, like I said, the one phase that the cells spend the most time in, and that is interphase. So it's the busiest part of the cell cycle, and that's because the cells are doing all of their daily functions that we need them to do to survive. So they're making energy in ATP, they're repairing themselves in other parts of the body, they're excreting waste, and they're manufacturing new proteins and organelles. It also goes through replication during interphase. And the replication is the duplicating or copying of your chromosomes. There are 3 parts of the interphase itself. So G1 phase is the cell will actually double in size. S phase, is where the DNA itself will replicate. And then G2 phase is rapid growth. So this is where it makes enzymes, proteins, and structures needed to allow the cells to divide into 2 separate cells. So they have to go through all 3 parts of this before starting mitosis. So now let's talk about what happens during mitosis. So the biggest thing is the nucleus will divide. And there are 4 phases of mitosis. So interphase has 3 of its own. And then the 4 phases of mitosis, which are the ones that are more commonly known, are prophase, metaphase, anaphase, and telophase. So first we'll start with prophase. And this is the longest part of mitosis because this is where everything basically has to line up to cause cell division to occur. And that's a struggle. Cells may be small, but the things inside of them are even smaller, so it takes them a long time to get where they need to go. So the chromatin or chromatin will coil up into the chromosomes. So it's this loose thread-like structure that we've discussed, and it's going to form into those X's that you saw, that you can see also right here. It's going to form into these X's. The genes go from stringy chromatin to condensed X-shaped chromosomes. And then those chromosomes are composed of 2 halves of sister chromatids connected at that centromere. The nuclear membrane of the cell will also disappear. So remember, that's the part around the nucleus. And this is actually what will allow the chromosomes to be accessible to split for mitosis. And the centrioles will migrate to opposite poles, and this is only in animal cells. So if you look over here in this picture, these little like 3-legged things are the centrioles, and they will go to opposite sides of the cell because they're going to actually be used to pull those chromosomes apart. And then we have what are called spindles forming, and it's more of a football shape because they will be like lines that shoot across the entire cell. And that's what you'll see happening in metaphase. So the spindle fibers attach to the centromeres, which are the center of the chromosomes, basically like a hook and pulley. And the chromosomes will line up along the equator or the midline of the cell, as you can see happening in this picture. So I'll just let this kind of finish out real quick. You can see those spindles are attached to the centers of the chromosomes, and the chromosomes had lined up on the equator right through the middle of the cell. This will then lead to anaphase. And one thing we say with metaphase is we met in the middle for metaphase. So the chromosomes met in the middle. And then anaphase, the chromosomes are taken away. So sister chromatids are separated, and then they begin to move towards the poles. So whatever spindle they're connected to, they will then go towards that centriole. And that's on the end of the cell, which you can see here in this GIF. And one chromatid moves to each pole. Remember, they're two identical ones. And then in telophase is where we actually start to see the cells to almost form back into two separate cells. They don't separate completely yet, but you do see them start to. So the chromatids will uncoil. The nuclear membrane will start to reappear in each half of this giant cell. And then in the animal cells, so what our body goes through, the cell membrane will start to pinch together and divide the cell right in the middle. And in plant cells, the cell plate starts to form between the two, giving each one a cell wall. And then the last step to mitosis is cytokinesis, or the last step to the cell cycle is cytokinesis. And this is where the cytoplasm splits to actually form two separate cells. So telophase starts this process, and then cytokinesis finishes it out by actually splitting the cells. And for animal cells, the plasma membrane will finish pinching together and form two separate cells, almost like a giant case of exocytosis. Remember where the plasma membrane would kind of pinch together and exit something out of the cell? It's like a large form of that. And then in plant cells, that cell plate will form across the equator, causing two new plant cells to form. So our results for mitosis, you should have two identical daughter cells. So they are carbon copies of each other. Both have the same number of chromosomes as the mother cell, which is the original one that it came from. And then what controls the cell cycle? So we said earlier that enzymes have to be formed before this can happen. And that's because they monitor the cell's progress from phase to phase. So these are called your cyclins. And failure to produce may lead to cell death or uncontrolled cell division, which we'll talk about as a mistake in the cell cycle. Enzyme production is directed by the genes or the chromosomes, so they know what is needed to perform this task. And then remember that genes are segments of DNA that control the production of one protein. Another important control factor of cell cycle is contact inhibition, and this is where cells come in contact with each other and they touch each other. This is a way that they can communicate by producing and secreting chemical agents to know, how the cell production is going. So sometimes there are mistakes in this cycle, which you can see in this GIF here, and that this GIF is uncontrollable cell reproduction, and this is actually how cancer occurs in patients. So it's currently thought to be caused by changes in the gene controlling the production of enzymes, which is a mutation in the genes. These changes are expressed as cancer when environmental factors trigger damaged genes to become active and reproduce. They will form masses, which we know are called tumors. And then as they enter the circulatory system and spread, forming new tumors, that is metastasis. So if you've ever heard something has metastasized, that is this